Hey, Erin, you're on mute.
Thank you so much, Allison. My apologies. So welcome to our May edition of Clinical Research Connections. We'll just give everybody a moment to answer a couple of polls as we always do and get settled. And our first poll is, our first poll question is, what is your role in our research community? And it looks like we've got a pretty decent distribution this time. Sometimes we've only got, a couple of different roles, but it looks like we have folks in every role except for PI at this edition of Clinical Research Connections. And so I will follow that up with a question about longevity. How long have you been involved in clinical research here at the university? While everybody answers that, I will go ahead and go over our topics and speakers. Today we have Rebecca Baldermann, who is going to be speaking to us about the CTRC Core Lab. And we will also be hearing from Nichole Carlson from the Center for Innovative Design and Analysis, or CIDA. And then we will have some time for questions at the end of each speaker's presentation, but also we will make a little bit of time at the end of the presentations for the clinical research community to share any announcements or thoughts. So if you have some that you are thinking about and would like to share with the group, we will give you some time for that at the end. So taking a look at the results of our longevity poll, it looks like we again have a pretty decent spread with a strong lean towards the 5+ years or 3 to 5 years. But we've still got some people here who have been here for 0 to 6 months. So I'm glad that those of you who have been here for a little bit less time are aware of Clinical Research Connections. So tell your friends. So without further ado, I will go ahead and hand it off to our first speaker today. Rebecca Baldermann, please take it away.
All right, so I'm a little rusty on doing presentations by Zoom, so you guys will have to help me out a little bit. Can you see my screen at least?
We see your screen. We don't see the presentation view, but we do see your slide deck. There we go. Perfect.
Okay. And then let me just toggle for a second. All right, great. All right. So my name is Rebecca Baldermann. I'm the assistant director of the 2 CTRC labs here on campus. So since we have some longevity people here, the CTRC has actually evolved over time. We actually started as a GCRC, I think 40 or 50 years ago. I was not there. But yeah, so it's been an interesting journey, but just as of note for the people who have been here for about 5+ years, we used to actually function fairly independently on campus as 2 different CTRCs. One would be called the adult lab, one would be the children's lab. We decided that for more efficiency and communications, we thought might as well mesh together because we were working independently, but also trying to have different test menus. And so we had to work side by side anyway. So if you saw some changes, felt some changes, that would be kind of why. We had a few people retire as well. We also had our lab in Leprino that was called the adult lab. We moved that to the AHSB building. So we have the nice, pretty fancy new lab with a glass wall. So you can always come and spy on us. And that makes it really awkward. But yeah, so that's kind of some of the changes. And then also with that, we had some growing pains. So I'm sure some of you have felt that, and my apologies, but we should be out of the woods now. So thank you for your patience through all that. So to kind of give an overview of how the CTRC functions, I like to make the joke of we're basically like the 3 branches of the laboratory. So we have our CTRC lab that you guys know and love so well through the CCTSI, but actually we also function as a reference lab for diagnostic purposes for the Children's Hospital, and we are the National Research Center for the Biochemical Markers for the Cystic Fibrosis Foundation. So we're kind of functioning in all of these different roles at the same time. And then as I kind of spoke, we have two different locations. So we have our children's lab, and then we also have the children's lab in the basement, humorously enough. And then we have the Anschutz lab, in which we're on the 6th floor. And so we actually get some natural light. Our hours of operation are the same for both laboratories, so it's 8 to 4:30 Monday through Friday. We do follow the university schedule and snow closures. That kind of gets tricky for some people, especially with the UC Health nursing. They actually are on UC Health schedules, so they might be open, but the lab may not be. So that's something to kind of keep in mind when scheduling. We have 168 assays. Hurry up and read quick because we're going to have a quiz at the end. Just kidding. These are all available. Doesn't matter, like they're split between the two labs, but you can order them for any study, and it can be a combination. We move samples across all the time. And because we are research, we also can bring on new tests, but unfortunately it is at the funding by the grant, by the requester. So that's something to kind of keep in mind, to reach out just in case we could potentially bring it on for whatever studies you guys are working on. We are CAP accredited through CLIA. Because we have two physical locations, that also means we are CAP accredited by two different numbers, and we have two different medical directors. So the one at the AHSB, our medical director is Lisa Meyer. She actually spends most of her time at National Jewish, where there's also another CTRC there. And then also our children's location, where I am currently, the medical director is Scott Sagel. So these certificates are often asked by different monitors. These should be updated and always available online whenever you need them, but just be mindful of what assays we're supporting. Typically through this group, most of them you would just need the one by Lisa Meyer. Our website is cctsi.cu.anschutz.edu. You can go to resources, CTRC, pricing, and then you're basically in the lab, and so you can see all of our different assays. We are tweaking our website to try to make it a little bit more user-friendly to search by assay. Just a warning right now, it works by, it doesn't search all content, it's only by the beginning, so just kind of be mindful of that. What is nice is each one of these things is hyperlinked, and so that's what I'm showing here on the left. You can see that we have the price, the name of the assay, the units, what sample types are allowed, and so on and so forth. So this also helps not only for monitor questions, but also for manuscripts for any PI-initiated studies. Often they need to know method, sensitivity, and things like that. And so we do try to keep that on the website, and so that way it can always be at your fingertips. Just to put, actually, sorry, my screen has too many windows there. So just to put it out there, we do function by COMERB. We call it our Mecca. If you have any questions, if you reach out to us and you use one of your acronyms, we're going to say that's really creative, but I really need a COMERB number. One of my favorite examples is optimize seems to be a really, really common acronym used. And so therefore, when someone asks about optimize, we've seen 3 of those already in the past 6 years. So we really do request anything you ask for. If you need to put it for your own records, if you need to use your acronym, that's completely fine. But we do at least ask for some type of reference for that COMERB number. So, and especially we don't use the pharma name because that gets really confusing too. They get really creative with those too. So I'm basically going to take you through a day in the life of how this whole process works from the laboratory perspective. So obviously during startup, we have encore review. It's always helpful for us to have that lab manual as soon as possible, especially, I mean, this is specifically pertaining to any industry protocols because they all have their different nuances needed. Nursing orders will come next, and then I can generate lab slips when applicable. So this is only for those in PI studies. And then we'll have a PI meeting in which we can kind of hash out with the different departments, any logistics or anything that's needed. The scheduler gets involved, and then we can dive right in. So for the visits, the participant is seen, the sample is collected, and then it's dropped off with the lab with a lab slip. All samples need a lab slip, please, please, please. And then the sample is processed, and then the study team can pick it up once notified, or it depends on how the flow of the study is. Obviously, if you have to process same day and ship same day, you won't be notified. It'll just be waiting for you on the table, which I think most of you are already familiar with that process. So then I've actually had a couple of study teams just reach out with very good questions. And so I just stole them and took them verbatim. So I was going to go through a couple of these. So what is the general workflow of the core lab from receiving a sample to processing? So when the sample is dropped off with the lab requisition and aliquot tubes, the processor will note the collection time in Copia and we'll order it there. And so that way we have our billing and processing and documentation. Chain of custody obviously is very important to all studies and it impacts the lab as well. So we help with that chain of custody. We'll process that sample following the lab manual when applicable, and then we'll store it in the appropriate temperature, ambient, fridge, freezer. And then we actually also do keep that lab requisition and we file and save and scan later. So how do we prioritize and balance it all, basically, is where this is coming into. We do try to, you know, I mean, it's kind of just how the lab functions. So we'll bring in more people if need be, like from different machines, and say, hey, can you pause and help me out here? We enable more centrifuges, and we just prioritize based on the collection date and time and what's needed. So this is kind of off of the same thing. How do we keep them all straight? Obviously, we have over 100 protocols a month that we're supporting, and those even rotate out. I think we have 300 currently active and open, so any of those can come in at any point. We do have those 8 centrifuges, and that's one way actually to segregate protocols as they come in. And then we can even separate by different colored racks, benches, tube types, things like that. So we have different methods and color code systems to really try to keep different protocols straight. It really does come, though, back down to that lab slip to make sure if it's a de-identified sample, we can cross-check it versus the nursing epic label versus the de-identified on the lab slip versus what's actually on the tube. So those pieces and elements are really critical for our success. And then how can coordinators reduce the burden, but not making some for themselves? So it's always help me, help you. So I would always suggest if you have any unique materials, just go ahead and drop those off ahead of time. There are some that have like special reagents, or if anyone knows about cryopreservation, sometimes having a dedicated Mr. Frosty can be helpful. Make sure that lab slip is always complete, tubes are labeled, and if there's, if the participant is not in scheduler, which we do have sometimes if we don't have the participant seen at the CTRC, it is nice to have the lab be aware of it in advance. And so we know that a bigger study with multiple time points is coming. Are there any common pitfalls that we should be careful of? I keep coming back to it. Lab slip, lab slip, lab slip, making sure that that's complete. Have that PID on there as much as possible. Check box tubes, like make sure everything that you have is included, and then making sure that all of those supplies that we need are dropped off. One thing also of note is one thing we've noticed is with some of these bigger studies, especially with time points, we'll just get a bag of aliquots. If you guys are having to actually label those aliquots, if you could somehow organize them in a box and drop them off in order in the box, if you're having to do that anyways, I think that would be really critical. Otherwise, we're dumping that bag and we're separating them before we can even get started. So something else maybe as a help. So to kind of go through the lab slip, a couple of things that we notice that are pretty common. This one you can see these are actual things that were dropped off. And so you can see right off the bat that timestamp is right on the label, so you can't even read it. The visit's not even circled, so we're not sure if it's visit 1 or 2. So we sometimes will have to look up in scheduler, but even sometimes that it's not there. So those little things are pretty helpful. But yeah, you can see with this one, everything is very legible. One catch with this one, you can see on this one, I marked off obviously the epic label, but obviously the epic label is going to be John Henry or whatever. But then you can see the tube that was dropped off is de-identified. So now I have no way of really knowing that this lab slip is associated with this tube because one is de-identified and one isn't. So those are different things to look at. But you can see this one is wonderful in the sense of you have your sample collection date and time, you have your PID there, you have your blood versus urine. You have one marked off because they didn't want microalbumin this time. So that's something else to keep in mind is it's okay if you need to modify for one particular visit. You can just mark it off. If it's just a complete change in visit for the entirety of the study, though, we do ask that you contact us so we can update that lab slip. Now the tube itself, so this is a wonderful one. We call it the window. So in this one, you can see not only that it has an additive in it, handwritten, but I also can see that sample level. So that's really helpful. When you wrap the tube all the way or the label all the way around, I can't see when I spin it where my crit begins and my plasma starts. Also, the pen is very important. So you can see on this one, this was dropped off at us. We could not figure out who this was. So by process of elimination, we could kind of figure it out, but we had to talk to the study team to have verification that that's actually what it was. So you can see pen matters. And then this is this is obviously not a picture of our freezer because we don't store food in there. But yeah, so you can, for anyone who has dealt with doing different industry studies, you know that that's kind of a hot mess in there. There's kind of no real way we can fix that except for, you know, having people come same day. Because we have so many protocols, we kind of can't really, we can't have a box per protocol. We would have 300 boxes in there in a heartbeat, and we just don't have room for that. But definitely know your kit. One unfortunate piece that we have had is people will drop off urine, for example, that's supposed to be stored at in the refrigerator, and they are not aware, and it's just sitting in the refrigerator. And the problem with that is it's so hyper de-identified, I can't even track or trace back to a date or a protocol or anything. So please do know your studies and know your temperatures. And so that way you make sure that you get all of your samples at the end of the day. And then how long should you wait before you pick up your samples? It all depends so much on how busy the day is and also how extensive your study is. So we do ask that you just, if you're on a tight timeline, you can definitely bring that up with whoever's on spin to just say, hey, I need to get this out by 3. What is your timeline? Things like that. And obviously we can give you a pretty good estimate. Finally, my own little other piece is that this is a shared shipment space. So this is actually pictures that I took fairly recently of how this area looked. Just of note, I can't help but put it out. There is actually a mask. If you look in the left-hand picture, there is a used mask that someone just left. I have found used gloves. It's pretty, it can get pretty bad. So I do kind of want to just remind people to use this space as a shared space and kind of be mindful of other people who are having to use this space. And then also it is a laboratory space. We've had many people ask for access into the lab, and unfortunately we just simply can't. We have biohazardous materials in there, but also we have radioactive materials as well. So that's the unfortunate piece is you would have to go through complete radiation training, and then it's an annual training and an exam, and it's a whole thing. But the biggest part also of why we really had to pull people back is we had under numerous occasions people storing shipping materials like ice packs in our freezers next to radioactive materials. So we are asking that people stop storing things in there because we need to be mindful of the type of contents in there. So we do apologize for the inconvenience. We have been sourcing different options on where we could potentially move this space out of the laboratory space so you could still access what you need to, but then won't be limited on time. So we're still assessing that overall, but hopefully stay tuned for something a little more efficient than that. And otherwise, I think that's pretty much it, unless anyone has questions now.
Thank you so much, Rebecca. And folks, please feel free to share your questions in the chat as well. Besides what you mentioned, is there anything else that the coordinators or supervisors who are training new coordinators can do to help them better engage with the core lab, or just make things easier and make sure things run smoothly?
I think whenever you have any information, don't hesitate to share. I'd rather be given way too much information way too soon than way too late. So if you have a new lab manual, just please forward it as soon as possible, or even just stop by and say hi and kind of see where everything is. And yeah, yeah, I would just engage with us. So so it looks like
Thank you. I have a question from Jessica Scott in the chat. Rebecca, are we expected to provide our own aliquot tubes, or can we use what the lab has on hand?
Yeah, so when I refer to those aliquot tubes being dropped off, that's typically for industry studies in which they're very particular on what barcoding and things like that. If you're just if it's an investigator-initiated study, we take care of all of that. You just drop off the collection tube, and then, yeah, we have our own barcoding system, and we can do the assays and everything. Yep. It's very hard to kind of jump back and forth and differentiate between the two, but still answer all the questions.
Excellent. Thank you. Any other questions from the group? All right. Well, thank you so much, Rebecca. I really appreciate that. That was a very helpful presentation. So we will go ahead and move on to our next speaker. Dr. Nichole Carlson is going to be speaking to us about the Center for Innovative Design and Analysis, or CIDA.
Great, thanks. I'll share my screen here.
Thank you.
CIDA is a campus-wide biostatistics and data science resource, and we also do primary research. And so I'll kind of go over what the role of this center is and a little bit about biostatistics and its interface with data science. Our goal is to help people build teams that enhance their research. So we're really big on team formation and embedded members. So I wanted to highlight, because of all the different names that have come out, biostatistics, bioinformatics, data science, AI, what is biostatistics? Because there's a lot of overlap. We tend to focus on the intersection of statistics, biomedical health, and computer science. And I think in this talk, I share other bubbles. I'm not sure if I was trying to be short or not, but so there's lots of 3 overlapping Venn diagrams for all these different areas. We really focused on the application of statistical techniques in health-related fields and developing new tools to study these areas. And we really spent a lot of our time thinking about study design issues, sources of noise and variation, and trying to figure out how to answer questions without getting trapped with overinterpretation of patterns in the data. And so that's what this quote here is about, is how do we formulate that? I just highlight, we do focus a lot on study design, which a lot of the other areas focus a little bit more on the data collection or the data analysis side. My personal philosophy, which I bring to this group, is that biostatistics is fundamental to all research. So it doesn't matter if you're doing basic science or trying to work with our electronic health record, or you are working with mice, that we can help enhance that whatever the research is, and that it's very costly to try to do it alone. And so there's some very famous stories about like Duke's whole IRB was shut down over a decade ago for just the simple data management issue in an analysis. And so we really view in CIDA that the best projects are where the needs and goals of the work are matched with the expertise and goals of the biostatistician. So I spend a lot of time matchmaking. So I'm like dating for research. And if you work with researchers, which I think all of you do, my mantra, which came from someone at Wisconsin, is come early and often. The earlier that your team members are talking to a biostatistician or like-minded quantitative person, the more chance there is for a successful partnership. When you come a week before because you realize something on a grant deadline, it's very hard for us to make meaningful contributions at that time, though we'll usually say, hey, next time you do this, let's meet earlier, and then we can work on your next project together. So this is where I do have the 3 bubbles. This is what I think has become very confusing is almost everyone looks like they are emerging of statistics and computer science and some other aspects. Like I said, biostatistics really focuses more on study design. It is not only clinical trials focused. We also act like bioinformatics and act like data scientists, so it's just confusing. The bioinformaticians on campus, I think, would tell you that they work mostly with molecular data and fairly straightforward designs, like 2-arm designs, a treated or an untreated group, or a case control. And once it gets past that, or even when it gets into human samples and they have to adjust for factors about the patient, those analyses are usually beyond their training. Data science typically has been not medical. And so the information that we bring when we partner with data science is really on how to work in a medical framework, work with confidential data. So, and just in case you also have looked up our new biomedical informatics department in medicine, you know, bioinformatics or biomedical informatics has bioinformatics in it, but many other disciplines too. And so my point on this slide is it's just very complex to wayfind. And what I tell people is if you know nothing, come to us, we'll help you. Even if we're not the right individual, we'll help you go to the Center for Health AI or connect with the bioinformatics team if you need more molecular expertise than our members have. So I'm going to skip over this in the interest of time. So the other thing I talk to people a lot about in CIDA is we will help anyone from a simple consulting or a 1-hour consult all the way through true partnership and team science where we're co-writing grants together, sharing in first authorship. And so when I meet with a group and we're thinking about partnership, I ask them a lot of questions about what type of relationship do you need or desire? And so if we're meeting with a new person, then I'm talking to them, is this something that you're just you have a small project, or it's your first project, and you're not gonna write any grants, you're gonna write a few papers in your career. Maybe we're just in a consulting role with you. Maybe we're gonna co-author 1 publication or make reports for your team. We spend most of our time in the research collaboration space where both groups are bringing together specialized expertise, working together repeatedly, getting to know each other, getting to know the members of the team. And in that role, we serve as co-investigators on grants. We develop subject area knowledge, and we work together usually for a long time. Several of these then in our career will turn into team science relationships where we're really truly acting as equal partners in the relationship. And I'm on grants where I'm a multi-PI. I am writing both clinical papers and biostatistics papers. And we're really just truly sharing in the whole scientific endeavor. But we have relationships across this whole spectrum. And I think it's really valuable when we're all in alignment. Where we sense tension in teams is when like our team member thinks, oh, I'm supposed to be writing grants and stuff in this team, and they're like, we just need you to get these papers done over here, and they're like, I'm not valued. Well, it's not that they're not valued, it's that they're in the understanding of what each person is doing for the team isn't clear. And so we have a consulting center. You'll see a bubble diagram later where I talk about all the collaborations we have across campus. And then at the end, which I don't think I'll have time today, we have some team science stories. And so these are questions which I can share these slides for you guys to have, that if you are working with a new researcher who doesn't have biostatistics, or you're with a department or a group that doesn't have any relationship with us, these are the types of questions I ask people to be thinking about to help identify what are your goals in having a relationship. And so the challenge with our campuses is we are not the only group that does this. I'll tell you there's 5 or 6 groups that act exactly like us. We're the biggest enterprise because we're the campus-wide resource. But we also interact with Cancer Center biostatistics shared resource, children's, if you do child health research, your point of connection would be the Research Institute through Children's Hospital, Accords, which is a adult and child health outcomes research focused group, has its own biostatistics core. And then we're tightly affiliated with the Department of Biostatistics and Informatics. All of our members are members of that department, but there are people who aren't members of CIDA just by the nature of how they were hired in their position. I think the point of this slide is to remind us we're totally decentralized campus, and it can be really frustrating for you guys when somebody walks in. At the end, I can bring up our new website. On our website, there's a new navigator system called the Cube, CUBE, and that once a week, all of these groups get together and talk about requests that came in and try to help that individual wayfind without making that individual go to each group. And so we are trying to kind of combat the issues that happen with a decentralized campus by working together on behalf of especially new individuals who have no formed relationship yet, and get them to the right home without them having to do all of the emailing that normally has been happening. I already highlighted that we're interconnected, and I just want to recognize for you all again that this can be a really frustrating system, and so I'm hopeful that this Cube link, which there should be an announcement out of the Vice Chancellor for Research's office next month in their newsletter. It's gone out to the Cancer Center. CCTSI will send it up to you, that we're all working together such that there's one link that you can put in a request to, and then we will get back to you with the human connection that you're probably desiring, and with the person who's probably going to say, yes, we can work together. So what do we do? And truly all the other groups do this too. It's just which area of specialization they work in. So we do a ton of facilitation. So we help people navigate the campus. We do 1-hour consults. We get about 3 or 4 requests each week just to help figure out a standard statistical questions, or should I get somebody more on my team? We write several hundred grants a year with the campus, and work all the way through the aims and the analysis plan, and then we put FTE in all of the grants so that we can support the work if the grant is funded. And then we do comprehensive study design and analysis for both short and long-term projects for just one-off things. So I don't want people to be deterred, especially when they're junior. They are just starting out. They might have 1 or 2 things or a small amount of startup money. Our consulting center will help them with whatever resources they have available to meet their goals for that project. Our bread and butter over the last 5 years, which some of you have seen me develop who are on this meeting, is team formation. And so this is where I hope most groups want to end up, is an embedded relationship where there's a couple people who have their academic home or their primary home in CIDA, but they do most of their work with another group, and they co-author papers, write grants, understand the nature of the science in the area, and work together for a long time. So we do about 22 dozen or so of these each year across the campus. And then with the support of the CTSI through its biostats program, which I also run, we do a whole bunch of 6-week short courses and do a week-long data week where we introduce people to software tools and other data management systems. And so that will come again, probably in January or early February. So when we're not busy doing all of this stuff, many of us, especially our tenure track faculty and assistant professors and above, do innovative methods research. So I work with lung imaging data, actually with Lisa Meyer, who was named in the previous talk, on how to integrate clinical omics and imaging data together. But we have a wide variety of people across our 20 PhDs in methodology. So if you have a problem and you don't know what to do with it, there is probably a person here who's interested in figuring out a new approach. So who are we? Right now, we're about 20 master's faculty and 20 PhD faculty. And then we have a dozen graduate student trainees all getting their master's or PhD in biostatistics. And they're, as you see by this photo, I love it, they're energetic and happy people and love to collaborate. And we tend to hire extroverted biostatisticians, probably because I'm the most extroverted biostatistician in the world, that they love to help people. And it's a pleasure to watch them be energized to like work with new data and work with new people all the time. And so we're very broad. This is not even an exhaustive list. And then these are all the different departments and units that we're working with. So it's probably too tiny of font for you to see, but if you are curious, if your group is in there, you could write me after and say, does our group have an agreement? You may not even know. And so this is what I saw. Allison Lincoln was on this. She's watched me build out from about 3 of these 6 years ago to all of these. And we still are talking with different groups across the campus. We definitely have not exhausted all of the groups who don't yet have a biostats core in there with their unit. Okay, so what are the benefits? I will have a few. I think I have a minute or two. So what happens when you get embedded and move to team science? I just want to share some of our huge success stories. In collaboration with the CTSI, they got a very large grant to do real world effectiveness of monoclonal antibody therapy in COVID-19. And we needed to pull together a huge team. This is just part of our team on the bottom. And because we had CIDA with such a large body of individuals, we could repurpose our time on things that are high priority versus not high priority and build together a 4 FTE biostats team within 6 months. And so this was really a landmark thing for the NCATs at NIH. And I ended up directing this group of 3 that's on here, and really gave information back to the hospital and the nation and FDA on when monoclonal antibodies were working, when we started to see evidence that there was a shift in the variants and it wasn't working. And so it really was a partnership, and now almost every one of these individuals still works with the PI, which was Dick Gindy on this trial in emergency medicine. But because we were large enough, we could do something really big quickly. And so we're excited to help with other center grants and other large initiatives and use the breadth of experience we have to do larger projects than just individual R01 projects. And then pulmonary is our longest standing collaborative agreement, and it grew from this one gentleman on the left. His name's Ryan Peterson. They started out funding a half of his FTE now about 4 years ago. And with the success of the grants and the enterprise there, this is the entire team. They've gone into the bioinformatics space and have gotten more than a dozen grants funded. And so he gave me a slide of kind of how they process through their system. But I think the results to date are that they've, as of last year, had helped with 15 grant submissions over a year. They got 5 funded. They've had a dozen publications in a year on that team across a broad area. And so it's really helping their investigators advance their area of research quickly. And then hematology is our second longest collaboration. Diana Abbott runs that collaboration. And over time, her expertise has grown such that they've had 2 SCORE grants, and Diana's named the PI on a application for SPORE grants in cancer, and really have changed the way that AML is treated. And so it's been really fun to watch Diana develop the core relationships and actually have an impact in how patients are treated now. So I'm going to end there, and I can answer any questions. I think my summary about CIDA is it is a campus-wide resource. It's funded by the School of Medicine, and the dean before he left just secured our next 3 years of funding, which covers part of my time and has helped us grow to 50 people from about 10. And we're absolutely happy to talk with any groups that are looking to bring biostatistics into their team on a wide range of areas. So I'll stop there.
Excellent. Thank you so much, Dr. Carlson. If there are any questions from the group, please feel free to unmute yourself or share questions in the chat. I had one. Could you talk a little bit more about the team formation work and kind of research partnerships that that you do?
Yeah, so normally it starts with either a division head or a research head, or there's a core of faculty that have been coming to us, and then they say, why don't we have a biostatistician in our group? And or they'll say something like, we're thinking of hiring someone, could you help us? And I'm like, well, actually, we will hire the person and mentor them and help make sure that they have the right training. And so then we just we start to have a conversation. It's variable on where people are in the thought and the formation of the idea and whether they've even thought about resources. But we usually sit down and start talking about how much money are you thinking about putting into this, and what's the size of your group? So we usually try to at least get first of like, okay, we have $100,000 to give to this problem. And then I can come back with, here's the kind of volume of work that for that amount of money people can accomplish. Is that in line with, you have 24 faculty all trying to write grants each year, or 6? And so, but I usually say before we meet, why don't you think about how many research faculty you have, how many clinical scholars you have who maybe won't be developing their own lab, but need a couple papers to show scholarship and state of the art, remaining state of the art in their area, trainees. And then once they give me those numbers plus some rough ballpark of resources, which can be variable, right? Sometimes they come back and they say, oh, we thought we could do this all for 20,000, but now that you've come back, we realize we are going to need to put aside more money. And then we sign in, we will either hire individual together and they'll interview them. And they may only work part-time. Most of them work part-time, like half-time or so for a group, and then they'll have one other group. Or they'll do like Ryan did, where Ryan's time has stayed about the same, but they've been bringing on new team members and they help co-hire and interview those. But we sign an agreement, so we're kind of all in the same place of expectation. And then we do, with whatever that team is that brought us together, whoever signed it, we'll do kind of an annual check-in. The individuals that are on the agreement, we have some member, at least a minimum of one person, send in something for their annual evaluation on how this is going, but sometimes they'll write lots of people from the group to send in information to us, and we'll take it all. Because most of it's positive, right? If they're asking the person to give an annual review, but it's generally a process, and it can range over half a year to several years that we go back and forth of where are you guys at, or we're doing a hire now. Are you ready? Are you guys moving along? So I don't know if that's helpful, but there's just a lot of conversation going back and forth about what people want, and then where does it fit into their plan for their research in their unit.
Thank you. That is really helpful. I have a question in the chat. Could you share the web address for CIDA in the chat?
Yes.
Perfect. Thank you very much.
CIDA .edu. We have a shorthand. And on there, one of the first buttons is this cube. So if you have a person in your office saying, I think I need data people, they don't even know what to call it, data people, you could just send them to this website and say, click the cube button. You might remember Rubik's Cube from it. Click it, and that group will help you get started. Because the team formation is not at the individual level. The individual level is more like I'm writing a grant, and they find us because they're writing an individual grant. Mm-hmm. And we do that. And we'll just talk with them about how that process works at an individual level. So don't be afraid of, I don't know how many, I can't remember your survey. I don't know how many of you guys work on the more administrative side with person where you might just find out about it on their grant and you look at their budget and go, there is no one to deal with this volume of data they're doing. You could send them to us then. And if it's too close to their grant this time, then we would say, hey, let's work on your next one together. We wouldn't, like, we'll explain to them, we really can't do anything meaningful now, but now we know each other, and so come earlier next time and we'll work on your next endeavor, because you're probably certainly not writing only one grant.
So that kind of leads me into my next question. What is kind of the minimum lead time that folks should give you to get to get in touch?
We love to hear from people 2 months or more in advance, and there's usually, the aims might say something like, I'm going to cure cancer. So we know they're not going to be specific, but at least then the person who's starting this brand new partnership on our side is planning their time, and they have an opportunity to really talk about your design versus coming with a month before is almost impossible. By the time that, because the grant's going to route in about 2 weeks after that, and that's about how long it's probably going to take your schedule to line up with another busy person's schedule. And so that gives us a few weeks to get together, learn the design, be able to feel comfortable to say something like, I think you might want to consider something different here. Or did you think about this? Or I don't have enough of the variables to do the power calculation or the right papers. And we don't even mind hearing earlier, 3 months, like the idea is a seed in your head, or you just click the administrative button in your own unit that says, by the way, I'm going in for June and it's, the end of March. We're happy that you just fill out a form there and say, actually, I have nothing to share with you now, except for I should have a consult. But once it gets past 2 months, it starts to get sketchy, which I write my own grants. That feels impossible to me sometimes. I'm like, I barely have the idea formed. What are they talking about? But we're okay having a lot of vague conversations and may deliver 2 weeks before a power calculation to you, but we'll have had time to talk and then do, I call it meaningful work rather than something that might get more reviewer criticism. So thanks for asking that. I didn't put that in. And we're the CUBE program, we just did it today. We're getting kind of forceful of we're kind of agreeing across all the groups that if you come in 2 weeks before, we're all going to say no, so that we enculturate that you can't wait till the last minute, that none of us will cut a favor, because we like people. That's what we do. So we just declined the first one and in our CUBE group where we said no, but next time come 2 months in advance. And these are the groups that would have been able to help out.
Excellent. Thank you. That is really helpful to know. And thank you so much for sharing about the Center for Innovative Design and Analysis, because it's a great resource. So again, thank you to our speakers. As promised, I will open it up for announcements from anybody in the community. Does anybody have any upcoming events, thoughts, notes to share with your colleagues in clinical research? Feel free to unmute, or if you want to put something in the chat, you are more than welcome to as well, and I will read it out. For our part, the next Clinical Research Connections meeting will be held on June 18th from 12 to 1 p.m. We will be hearing from the Blood Bank and from Encore at that session. You can find the registration link for upcoming sessions on the Cross Clinical Research Operations and Support website, which is in the chat. We will have a recording of this, like any other clinical research connection session, posted on the Cross website by the end of the week-ish. And if you have any ideas for future research topics, please go ahead and email researchstudies@cuanschutz.edu. Any other? The presentation recording will be shared. It'll be shared on our YouTube channel, which will be linked to from the Cross website. Any other announcements from the community if not, I will go ahead and just bid everybody a wonderful rest of your afternoon. And thank you so much for attending Clinical Research Connections. Thanks. Bye, everyone.